Here, I would like to share with you how our platform can help the scientist through 3 case studies. In this first case study, one of our clients was interested to discover a patentable gene from their own private microbial collections. However, there are many challenges. Number one, data privacy that Nasir just talked about. So due to privacy, organizations can't use their private data for query to the public databases. This is a major roadblock, especially for industry clients. So number two is creating and mining the patent database. So you need someone who can create for you the special patent database so that you can use. Number three, data resources over many repositories, both within and outside of organizations. So data usually kept in multiple repositories and are not properly maintained both within and outside of the organizations. And number four is most important, is the availability of the bioinformation. Suppose even the scientists settle for command line based tools and the mining the data with the help of the bioinformation, but the availability of the bioinformation time is a major challenge because bioinformation help multiple other projects and they work for the entire R&D team. So getting in time, the bioinformation time is very important and it's often don't happen. So as a quick note, for this case study, we are using the data from NCBI and not from any of our clients. So as you all know, in a typical workflow, the scientists prepare the microbial samples from their biological library collections and submit them to the NGS facilities for sequencing purpose. Once the raw data are assembled and annotated by the bioinformatician and store them on an internal servers and various other devices generally in raw format. And then scientists will mine this data for microbes containing pesticidal genes with the help of the bioinformatician. And then they conduct downstream lab work before testing it in the field. So in this typical workflow, according to the scientists who worked on them, and also as per the NSF-sponsored study that we conducted earlier, this data access and mining step is one of the time-consuming part for the scientist, as often bioinformaticians are not available due to their busy scheduling, and also they support multiple other projects in the organizations. And I, being myself a bioinformatician, I know that this data access and mining part is one of the most important part in this total workflow. And it is also very painful process or step in the whole workflow. So if you want to hire a bioinformatician for each process, for each data processing step, or each project, then it is also very costly affair for the organizations and the institute. So here, Loci fits in very well in the data access and mining stage of your workflow by being it on-premise or on your cloud server. Loci serves as a central repository. Nasir already talked about this. By unifying all your data in one place and connect the data to the mining process, analysis, and visualizations. You can do it in just a few clicks and thus free up the bioinformatics time. And also any scientist can also do by themselves. So Loci being a web-based platform, scientists can access the Loci using any modern browsers on any devices, and after logging into their secure environment, they can see the home page and the dashboard, which showcase the data they have and how they are organized. So in this study purpose, we downloaded the whole genome sequence data of about 135,000 bacteria from NCBI and arranged them into two major genome collections, namely public and private. So if you see the donut, the red one is the public and the orange one is the private. So in private group, we created two organism-specific sub-collections for BT and SpinoSub. And I must say, this page is very much customizable and flexible. So you can store the data by projects or by organisms, etc. So all the flexibility is with you. And depending on the kind of projects or what you are looking for, you can add these data sets. Now to proceed with this project goal, click on the genes in the menu indicated by the hand symbol. on this navigation bar, and that will take you to the dedicated genes page. So after coming to the genes page, first question to ask, how many pesticidal genes are there in their private collections? So on Loci, you have the flexibility to search in all or specific collections. In this case, the scientist selects the private collections and search for all genes under the pesticidal descriptions. Now, this search resulted in 2 pesticidal genes out of 3 in private collections. You can also download this sequence in FASTA format from the right-hand corner drop-down menu, as you can see in this screen. Now, the scientist has discovered that there are 2 pesticidal genes in their private collection. The second question to ask, are these genes patentable? To find the answer, click on the blast menu indicated by hand symbol on the navigation bar, and that will lead to the dedicated blast page. Now paste this FASTA sequence of both CRY genes and select the NCBI patent nucleotide sequences for multi-blast analysis. And for this pre-formatted database, we have downloaded all the available 99,000 patent sequences from NCBI. But as you know, that some of our clients actually mentioned that they would purchase additional patent database from various vendors. So if you have that additional database, patent database, then we can do the pre-formatted database, which you can keep on the Loci along with the NCBI data. And that will broaden your search horizon. And creating of this pre-formatted database is totally customizable and client specific. So we, according to your need, we can totally customize this pre-formatted database. So here scientists also perform single, multi, and custom blast job using the combinations of different databases on Loci. You have to select the appropriate program and name the job for future references, and then click the blast. It will lead to the My Task page. This My Task page is unique, and it serves as a user-specific repository for all previous studies that the scientist conducted. So when you will come back later, and scientists can search for all the previous study that they have done, and thereby you don't need to run the blast job again if you already run it before, and thereby you can save a lot of time and energy for the organizations. Now, to see the results for a specific blast job, you click on here, and for these purposes you have the second one, and then you click here, and that link will lead to the visualization page. The results indicate that CRY1BA gene has already been patented, and also give the patent number, percentage identity, and other relevant information. Now, to discover the patentability of CRY5AA gene, click on the scroll down button and select the CRY5AA. These results indicate that there is no patent sequence that showed homology to CRY5AA. So as a quick note, this analysis is done using 99,000 patent sequence from NCBI, but typically the patent search will be much broader, as I described earlier. So you need some proprietary patent database from vendors. And if you have that additional patent sequence, then you can perform those patent search through the Loci as well. Now, to determine the novelty of CRY5AA, run a blast against all the public database on Loci by clicking the appropriate boxes and then do the blast. And these results indicate that the gene is showing homology to the whole genome sequence data of bacteria, where this gene is originated from. But basically, this is a self-blast test, so you're supposed to expect that kind of homology. But with this study, you can see what are the objectives we achieved. So here, Loci helped them discover that there are 2 pesticidal genes in their private collections. So out of these 2 genes, one of them is patentable, and it is a novel candidate gene, which I guess is a very valuable information for the scientists. And this discovery will help them to decide whether to allocate and prioritize lab resources for both genes, or 1G for all the downstream lab analysis. And as you see, all these discoveries are made in less than 15 clicks. So in this case study, we showcased for 1G. Now imagine if a scientist is analyzing hundreds or thousands of genes for getting the novelty and patentability information. Considering the challenges that we mentioned earlier, Loci can save weeks and months of scientist data mining time. And so they can start conducting their downstream lab and fieldwork quicker and thus accelerating innovation.